My name is Susan Aitken. I am currently working as a PhD student on the Arta program, which aims to develop an affordable HIV drug resistance test for use in Africa. I am a South African, born and raised. Coming from Africa, you see the problem of HIV every day. And you want to help. You know people who are suffering from HIV, you know their families, you know how it affects them. And you see when their drugs fail and when they get sick and how terrible it can be. HIV itself is a retrovirus, and a characteristic of retroviruses is their mass production. They replicate very quickly. They believe in quantity, not quality. So during their replication, they don't check themselves for errors. Sometimes one of those mutations gives the virus an advantage. And once that virus has an advantage, it is now a drug-resistant virus. And then the drug that is being used against that virus doesn't work, and the virus keeps growing. And we need to know what mutation happened in order to change the medication for that person. The goal of Arta was to develop an affordable resistance test for Africa. We wanted to get a test that is very simple, that is cost-effective, and that can be used in any lab within Africa. Commercial drug resistance tests require plasma. Plasma is a proportion of our blood. So in order to obtain the plasma, you need venipuncture. So someone needs to draw blood from the patient. It needs to be stored at -80. You need big -80 freezers. They need continual power, because if the power goes out, your samples get compromised. This is why nowadays for studies within Africa, it's becoming more popular to use what are called dried blood spots. For such a thing, we use a collection device. So they make a small prick on the finger, you get some blood, and you put it directly on the card. And that can then be sent to the lab for testing. So we can just post them. There's no need for -80, there's no dry ice shipment of infectious plasma samples. It's a lot safer for everyone, and it's a lot cheaper and quicker. What we have contributed as part of Arta is a more in-depth look at the samples themselves. We look at defining better criteria for storage, how long you can store the samples for, in order to formulate some guidelines to standardize the procedure, to ensure there's complete sample integrity, so that once we've performed the test, the results we get are a true reflection of what is going on. The research that we did was to investigate the effect of temperature and time on samples. We found that storage at room temperature should not be for more than 2 weeks, after which we should store it at -20. And within Africa, it's a reasonable concept. If you have a collection site in a very rural village, collect the sample, and within 2 weeks have it at a laboratory, maybe a central laboratory at one of the bigger cities. It has a lot of promise, but we need to make sure that the criteria are very strict, so that we're providing answers to diagnostics that are equivalent to the gold standard.